Okay, so I'm Steve Harding, and besides being co-organizer of the MIBIO 2023 meeting, I'm also chairman of the Royal Society of Chemistry Felix Franks Biotechnology Award Committee. And Felix Franks was formerly professor here at the university, but he was also distinguished chairman of the biotechnology group of the Royal Society of Chemistry for many years, and became well known in the field of chemical biotechnology, particularly for his work on carbohydrates and water, freezing water. He became known as Water Franks. And thanks to the generosity of his family, who have agreed to provide the medals for the award. And these medals are for early career scientists. It's an annual award, and it's for an early career scientist who has made an outstanding contribution in the field of chemical biotechnology. This is the 5th year of the award, and as in previous years, we've had an outstanding collection of nominations that have been made. But one person stood above all the others, and that is our next speaker, Chiara Arno, particularly for her outstanding contributions in the field of materials and cell interactions. And amongst the most significant of her finds has been the development of a technique to precisely control the morphology and size of semicrystalline particles in water, which relates nicely to what Felix was involved with. And subsequent research has built on this in the biological applications of nanostructures that are being prepared by crystallization-driven self-assembly processes. And I'm building on her original idea. She's pioneered the concept of 2D nanoparticles as tissue adhesives. So here is the award here, but I'm not going to give Chiara this straight away because she has to sing for our supper, and she's going to tell us about some of this exciting work. So the title she's given us is Self-Assembly of Crystalline Polymers for Biological Applications. So Chiara, do you want to take control?
Thank you.
So you know the...
Yes.
Yeah, okay.
Great. Thanks a lot, Steve, for the great introduction. So, first of all, as Steve mentioned, I'd like to say a few words about Professor Felix Franks, and specifically about what we have as common points. So he was born in Berlin. I was actually born in Italy, not in Germany, but we're both foreign. We came to this country both as foreigners, so I'm sure we face the same challenges to start with. And also, we both did a PhD in London. He did it in physical chemistry at the Berkeley College, and I did it in pharmaceutical chemistry at King's College. But what some of the common areas, and many of the common areas, are actually in the science. So by reading Felix Franks' publications, I found out that he's actually worked with polymers as cryopreservants, so to preserve, for example, in here plant tissue, but also biological structures. And this is actually some of the work we do in the group as well. So I have highlighted here an example of stuff that I'm not really going to go into depth and talk about today. But we look at polymers for single cell encapsulation. So what you're seeing here is a suspended cell surrounded by polymer matrix that in water. And this work has been done by Amaziah, who's a second-year PhD student in my group, and he's also in the audience. And as Steve mentioned as well, Felix Franks has done a lot of work in water and has published actually several books in water as matrix of life. And to the point that basically he got the nickname of Water Franks. And we love water too. So we do a lot of work in water. We try to make polymers directly in water, actually in PBS or cell culture as well, and in the presence of cells. And this is, again, a work that I'm not going to talk about today, but just to highlight, we developed a technique to form hydrogels in situ in the presence of cells using a technique of visible light polymerization. And this work was led by Alison Rigby, who's also in the audience today. So this directly links to what we do in our research group. So there's three main areas that we explore. The first one is about cell-based therapies and cell-based materials. And we use techniques such as cell surface polymerization, but also intracellular polymerization and bioorthogonal chemistries so that we can tailor specific groups at the cell surface without actually disrupting cell behavior. And as I said, we also do a lot of polymerization in aqueous media, which actually sounds quite straightforward, but it's really not. And the reason is that there's not a lot of monomers that you can polymerize that actually in water, or a lot of interesting one, let's say, that you can not only make a polymers with, but you can also functionalize afterwards. And so we look at developing new monomers and new chemistries so that we can polymerize in water and also functionalize in water. And the third aspect of what we do is in the field of biomaterials. So we work at developing hydrogels, nanomaterials as well of different size and shape. And we look at controlling, thank you. We look at controlling the size and the shape of these nanomaterials as well. And we use them as cell scaffolds, so to aid cell proliferation and cell differentiation as well. And this is mainly going to be the focus of my talk today. So first of all, why are we interested in nanoparticles of different morphologies, while most of you, I'm sure, are familiar with spherical nanoparticles? Nanoparticles of different morphologies, such as the cylindrical one here, or also two-dimensional platelet nanoparticles that you'll see later on in my presentation, are interesting for a variety of applications. For example, cylindrical nanoparticles have been shown to circulate in the body for longer compared to spherical nanoparticles, so they can offer a longer circulation time, so they're not prone to degradation as spherical ones. And also, platelet nanoparticles are two-dimensional, and so they have a larger surface area that they can get in contact with tissues or different materials. However, they're not easy to make. And this diagram here shows really that there is a very tiny window where we can access cylindrical nanoparticles of very controlled morphology. So it's relatively easy to get nanoparticles of different morphologies in one pot. So you get a mixture of spheres, cylinders, and so on. But it's not really easy to get just the cylinders to get specific control over morphology. And one of the techniques that is one of the greatest ones to achieve this is called crystallization DFSF assembly. So in this technique, we use semi-crystalline polymers that, as you can see here, have this very ordered and packed structure that distinguish them from amorphous polymers. And this packed structure allows them to assemble in two dimensions. Those are the platelets that I was talking about just earlier. This technique was pioneered by Professor Ian Manners in 2010. And what they found is that they, from one single crystallite, they can actually grow the nanoparticle in both dimensions. And the length of the nanoparticle can be controlled by how much polymer you add in. So if you add X amount of polymer, you're expecting the nanostructure to get to the specific size. And this is how you achieve control over the nanoparticle size. Also, they have enhanced stability compared to other spherical micelles or micelle structures made from non-crystalline polymers. They have a narrow, you can narrowly control the width and the dispersity. And also, and I'll show a little bit of this in my talk toward the end, you have the ability to modulate the nanoparticle surface chemistry. And so you have the ability to attach new functions to it and to create nanoparticles with multiple chemistries in it. So I'm going to tell you a few stories today, and the first one that I'm going to start with is this one here, where we use this polycaprolactone-based polymer. So polycaprolactone is a biocompatible and biodegradable polymer, and it's actually used in many devices approved by the FDA, for example. So it's approved for internal use. The other part of the polymer in blue is a polydimethylacrylamide, and that is a hydrophilic polymer. So this is an amphiphilic structure, so it has a hydrophobic component and a hydrophilic component, and it can self-assemble. And what you do with this polymer is to put it in ethanol for 3 hours, and you heat it up, and then you form very long structures, several micron long cylinders. So at this stage, what we have is a very good control over the nanoparticle morphology. We only get one type of structure, but it's not very good control in the terms of size, because the cylinders range from 1 micron to 10 microns in length. To achieve control of the size, we sonicate the cylinders to form what we call seeds. And the reason why we call them seeds is that we can then grow from them by adding a sequential amount of polymers in a controlled fashion. And so you can get any size that you want. However, as you can see here, we do this process in ethanol. But what I was really interested in is, can we actually do this in water so that we could use these nanoparticles for biological applications and to interface them with cells. However, no matter how hard we tried, what happened was that the nanoparticles were crashing if I transferred into water. And that is because water was actually getting in contact with the polycaprolactone core, so with the red side of this polymer here. And by getting in contact with it, it was disrupting the crystallinity of our polymer, of our nanoparticle, and the nanoparticle was collapsing. So to overcome this, what we did was to design a slightly different polymer. I put here for comparison the one that I was talking about before. So a slightly different polymer that has this polymethylmethacrylate block. And it's a very short block of only 20 units, but this is a very hydrophobic, very glassy polymer. And basically what it does between the hydrophobic component and the hydrophilic component is to protect the crystalline core from collapsing. And so in this case, the nanoparticles shouldn't actually collapse when transferred into water. So these can be self-assembled using the same conditions as I showed before. So you heat it in ethanol, then you sonicate to get your seeds. And differently from before, you can actually transfer the seeds in water, and then you can grow the polymer. You can grow the nanoparticle by adding sequential amount of polymers. And as you can see in this graph here, there is a very linear relationship between the amount of polymer we add and the size that we reach. So we get excellent control of a nanoparticle size. And what we showed as well was that by continuously adding polymer and growing these nanostructures, what we observed was that we can form a macromaterial, so a hydrogel in this case, that is simply formed by entanglement of nanoparticles. This hydrogel has self-healing properties, so you can crush it, and the hydrogel structure is disrupted, so it behaves more like a liquid. And then when you release the force, it comes together as a solid. And you can do that in a cycle, and so you can show that it's a self-healing material. And as we do everything in water now, we can actually encapsulate cells as well. So I'm showing here an example of 2 different cell lines. It's A549 is a human cancer lung cell line, and MC333 are pre-osteoblasts from mouse. And the viability was higher than 95% after 7 days of incubation. So we have a system that actually works well in water, and we can make materials with it. Another story that I want to show you, always in terms of nanoparticles and how we can use nanoparticles together with hydrogels, and it's this one here about using nanoparticles to reinforce allogeneic hydrogels, but also to impart new properties to the materials as well. So when I was working on this project, which was back in 2019, I came across this paper that was published in Nature a few years back, and they were using spherical silicon nanoparticles to glue together hydrogels and tissues. And they were claiming that basically nanoparticles would interact with the material and would glue the 2 blocks and would glue the tissue together. So I thought, can we exploit the ability that we have to make nanoparticles of different morphologies to actually tailor this property to increase, for example, adhesion or to make nanoparticles in tissue more adhesive together. And so the polymer that I designed was this one here. This has a polylactide core as opposed to polycaprolactone, as I showed before. Polylactide is, again, a biocompatible and biodegradable polymer, and it's a slightly different chemistry for the hydrophilic block. But the key point here is that we can use the same polymer, so we use the same chemistry to make 3 different morphologies, to make spheres, cylinders, and platelets. So any property that we see from the particle from now on, it's only because of the nanoparticle morphology and not because of the nanoparticles of its chemistry. So what we did was to see if any of these nanoparticles had adhesive properties, and we kind of repeated the experiment that I showed before in the Nature paper. We cut alginate hydrogels in different blocks, and then we put the nanoparticle in one block and tried to adhere the other one. And what you're seeing here is actually a rainbow gel made from different blocks of hydrogel built, glued together and suspended between 2 vials. And you can also pick up the material, and as you can see, the adhesion is actually quite stable. Obviously, no adhesion is observed if no nanoparticles are used. And what you can see in these graphs in here is that the 2D platelets have actually a much higher ability to adhere the hydrogel together compared to spherical nanoparticles and cylindrical nanoparticles. And we think that this is because they have a 2-dimensional structure. And so the 2-dimensional bits of the nanoparticles are allowed to interact better with the polymer at the top and at the bottom that they're in contact with, compared to any other nanoparticle that we tested. The next thing is that we needed to prove that this adhesion was stable in wet environments. And as you can see here, it's actually stable in cell culture media as well for up to 2 weeks. So we observed no detachment of the hydrogel, and we made also constructs encapsulating cells in one layer and with an acellular layer. So we built basically, again, our hydrogel with a cellular and non-cellular layers. And then we glued together 2 different cell types, as in this case, osteoblasts and chondrocytes. But also what we found out is that we could glue tissues together. And in this case, we tried with cartilage. And as you can see here, with the value of the shear strength, so the adhesive strength of these nanoparticles, they're actually comparable, if not better, with some of the materials that have been approved as adhesives for internal use, such as the PEG and the T-cell adhesives, so fibrin-based adhesives. So in the last few slides, as promised, what I want to tell you about is what control do we have on the surface chemistry of the nanoparticles and what we can actually do, what we can achieve by controlling the surface chemistry. And this is actually very recent work that had just been accepted last week. So in this case, what we are manipulating is the hydrophilic bit of the molecule, so the part that stays outside. And you can see as by changing the chemistry, we're changing the shape of the nanoparticles. For example, in here, when you use an aromatic ring with an heteroatom inside, as you can see in this picture here and this picture here, you can get actually more elongated structures compared to a more hexagonal structure when you use non-aromatic species. And the other thing you can do is also you can mix all this polymer together. Because of the ability that we have to add polymer to the nanoparticle, we can not just control the size in this way, but we can also control the surface chemistry composition. And in this case, we're building basically nanoparticles that have many different layers, and we have many different functionalities. And as you can see here in this picture, for example, with a confocal representative, we labeled only a few layers of our nanoparticle, and we know exactly what that polymer is inside our single structure. We cannot just manipulate the hydrophilic part of a nanoparticle, we can also manipulate the hydrophobic part. So I talked a lot about polycaprolactone and polylactide as a choice of polymer, but we can actually go to any polyester that we want. And in this case, what we did was to simply change the size of the ring that we're dealing with. And by changing the size of the ring, we also wanted to see if that has an effect on the nanoparticle morphology, but also if that has an effect on degradation of nanoparticle, because theoretically, if you go from a 6-member ring to a 12-member ring, you are delaying the degradation of your particles, so you can actually control that aspect as well. So as you can see here, we can build nanoparticles for using any of these polymers. And then in the next slide, I'm going to show you a little bit of the degradation work that we've done. So the first thing that is removed from nanoparticle is actually the hydrophilic part of it. So you get a degradation at this stage. This is, by the way, accelerated degradation conditions. So it's about 1 molar of potassium hydroxide we're using. So quite far from any in vivo simulation that we can get at this stage, but this is just a starting point. And as you can see, we first degrade the blue bit in here, which is the 6-member ring monomer that we were using. And then as we go, and that happens between the first 2 days. And then as we go far away, in about 13 days, we degrade also the 8-member ring of our, the 8-member ring monomer that we had in our polymer. And so we can control the degradation by controlling the nanoparticle composition. And you can study that very well by using AFM and looking at the height of the nanoparticle and how it goes from 15 nanometers to 9 nanometers. And finally, we can also control the nanoparticle in 3 dimensions. So in this case, what I'm showing is the ability to grow the nanoparticle hydrophilic corona in 3 dimensions. So once the nanoparticle is made, you can just add more polymer, and that will grow in 3D. And not only that, and that is because basically there is this group still present on the nanoparticle surface, and that's the group that will allow you to introduce more monomer and keep growing and keep polymerizing. And not only this, we have great control over the growth in 3D, as you can see here, so it's directly proportional to how much monomer we add. But also, we have a great control over the location of the growth as well. And so you can, for example, in a nanoparticle, select a specific region that you want to grow, and then you can grow just that specific region without touching the rest of it. So you have great control over the location that you introduce your functionality in. So finally, I'd like to thank my group members. So the fact that there's many names here and not for a lot of us in the picture means that we need a group picture very soon, a new one. A lot of my collaborators as well. So a lot of the work that I've shown today was done by Maria, Zai Zai, and Tian Lai, and founders as well, and obviously the Royal Society of Chemistry for inviting me today to give this talk. Thank you very much for your attention.
Thanks. Thank you, Chiara. That was absolutely fantastic. It really was. One question from me. So you've got this, with the nanoparticles, you've got 2 things. One is the chemical nature of them in terms of efficacy and things, and one is the physical morphology. And you hinted at something there. By changing aromatic groups and things, you could affect the morphology. Do these things work against each other, the need for a chemical delivery and a physical, or can they be controlled now? So we want something which is, say, cationic with some certain function, and we can produce that sort of size, or is it still dictated by the chemistry?
So it is and it isn't, in the sense that they can work against each other, as in sometimes you mix one polymer with another and you try to make nanoparticles with us and it just doesn't work, as in the plates would not grow, or you won't get a good control over morphology. But most of the time, actually, you're able to make it work, because we can have different layers in it. If 2 polymers are not compatible with each other, you can have a mid layer of a polymer that is compatible with the one that you have and with the one that you want to add, and so you can grow that this way. So the fact that we can grow multiple layers in it plays to our advantage in terms of going against polymer non-compatibilities sometimes. Yeah. And
in any case, this is sort of part of the research. You're trying to sort of look at more possibilities there to make it happen. So, yeah.
There's endless possibilities.
Okay.
Yeah.
There's time for 1 or 2 other questions for Chiara, if anyone has anything else to ask.
We try to encapsulate drugs in simple drugs or the peptides or anything like that within structure at the moment?
That's a great question, and that's actually what my other PhD student, Hannah, is trying to do. So encapsulation wouldn't work that well because, again, with encapsulation, you disrupt the crystallinity, and so you lose control over the nanoparticle properties. But what you can do is work with functional polymers. So instead of the boring and just alkyl polycaprolactone structure that I showed, you can have pendant groups in there that you can covalently attach drugs to it through, for example, linkers that then can degrade in vivo, such as ester linkers or amides or so on. And then you can create a nanoparticle that way, and then you can create different morphologies, and you have great control over it. And then you can degrade that selectively and release basically the drug that is attached. So yeah, you can do it, but not through encapsulation, but through covalently attaching it to the to the part of the polymer. We try just covalence for now, no host gas, yes. But yeah.
Okay, thanks. There was a question from over here. Yes?
Right, yeah. So it's a great question because it took me like 3 months to get nice pictures of those nanoparticles back in time. And the reason is that, you're right, there's a different contrast because one sample, the ethanol sample, you can image that a lot better than the water samples. Water samples are a lot harder to image to TM. So the actual staining is the same. It's uranyl acetate that we use. But actually, those pictures, I think were taken, if I remember correctly, using a graphene oxide grade. So graphene oxide is the material that works best with nanoparticles in water compared to normal TEM grades that you would use for nanoparticles in ethanol, and you would just add the staining there. So that's the different contrast that you see, yeah.
Okay, any more questions?
No, so the light we use to grow the nanoparticles in 3 dimensions. So we can use light and we can use visible light to achieve that. But to grow them, to actually control where it goes in the nanostructure by layer addition, you just basically do it by sequential addition of polymers. So you add polymer X, and then you add polymer Y, and so you know exactly where these are. Yes, that's done with light. So as I show you the group that we have on the polymer, so you can get a polymer, if we get a polymer without the group, so that's where basically the nanoparticle will grow in 3D only when that group is present. So if you do a patchy nanoparticle with polymer with the specific group and without, then wherever is that group, then that's where the nanoparticle will grow. Otherwise, when the group is not present, it won't grow. Yeah? Yeah.
So also does the residue impact the morphology of the nanoparticles?
Sorry?
The residue remaining, which is not bonded to the copolymers, whatever you have prepared, 3 polymers, like 3 blocks you have prepared.
Yeah.
There might be residues, because what we have seen that many times the residues impact the morphology of the nanoparticles.
So you mean polymer residues present in solution. So yeah, the nanoparticles are aged for a long time. Normally, it's about 2 weeks that you leave them aging to make sure that basically there's no polymer residues, there's no polymer that it's basically soluble in that, and everything is crystallized. If you have a semi-crystalline polymer, everything will crystallize at some point. So nothing can actually just remain in solution, especially with polycaprolactone and with polylactide. They are very, very good at crystallizing very, in a specific time frame of two weeks, as we've seen. So, yeah.
Okay. Well, I must say, Chiara, because I was a good friend of Felix, a good personal friend, and if you could have seen this, and he may well be from looking from up there, I don't know, you were absolutely thrilled by your presentation. So it gives you great pleasure on behalf of the biotechnology group and the Royal Society of Chemistry to present the 2023 Raw Scientific Chemistry Felix Franks Biotechnology Award to Maria, Chiara, Arno. Chiara?
Thank you.
Here you are.
Thank you very much.
Well done.